Microbiology is the study of microorganisms such as bacteria, viruses, fungi, and other microorganisms which are not visible to our naked eyes. Because these microorganisms are generally too small to be seen with our naked eyes, the science of microbiology is dependent on technology that can artificially enhance the capacity of our natural senses of perception. Microscope is used to visualize these microorganisms. Microscope produces magnified images of microorganisms, human cells and tissues and many other types of specimens too small to be observed with our naked eyes. Using a sterilized inoculation loop, a sample of bacteria called bacterial culture or bacterial colony is taken on the microscopic slide containing a drop of sterile water. The thin layer of bacteria placed on the slide is called bacterial smear. Some stains and dyes are used to add color to microbes so they can be better observed under microscope. Some dyes can be used on living microbes whereas others require that the specimen be fixed with heat before staining. This is called heat fixing. After staining procedure, the bacterial smear is observed under the microscope.